Alright, it's time to talk about the phage vector. So we've been talking about the different kinds of cloning vector that we use for molecular cloning, and one of such vector is phage vector, or basically the bacteriophage virus is used as a vector for the molecular cloning process. So what is this phage vector and what we mean by molecular cloning, and how do we use phage vector for the molecular cloning? So phage vector is basically lambda phage. Okay, there are different kinds of bacteriophage viruses that attack bacteria. So lambda phage is one of them. Lambda phage has its head region, it has its collar. This is the collar, this is the tail region. The head is actually composed of icosahedral symmetry made with protein structure, inside of which the genetic material DNA is placed. Then it has the tail region, and at the bottom has base plate, and the base plate has tail fibers with which it can be in contact with the surface, which is bacterial cell wall. So now we are going to talk about the use of bacteriophage as a vector. So basically phage vector means we are using bacteriophage as a vector because the bacteriophage itself can infect a bacteria. So now imagine this, the bacteriophage is a virus, it can attack a bacteria. So when it attacks the bacteria, it can deliver its own genetic content, that is DNA, inside the bacterial cell. So that thing, during the process, we can easily transfer or target DNA or gene of interest inside the bacterial cell with the help of the phage. So phage naturally is working as a delivery system for the gene. So phage is a natural delivery system for bacterial transformation or bacterial transformation events. Okay, so that's why you use it. But remember, there's a big catch. The catch is that the phage is a virus for bacteria. So phage will infect bacteria. It can infect bacteria, and as it can infect bacteria, it can cause diseases in bacteria, it can kill the bacteria. That is the biggest challenge to use phage as a gene delivery system. So what we need to do is simply we need to get rid of the this wrong effectivity, get rid of this nature of killing for the phage against the bacteria. We need to get rid of that. So we will make this phage a disarmed phage. Okay, so the phage will be carrying the target DNA, but it will lack the capability to cause infection, thus causing disease in a bacteria. That is really, really important. Okay, so lambda phage is a bacterial virus, as I mentioned earlier. It consists of icosahedral head and flexible tail. You can clearly see this is icosahedral head, this is flexible tail, and at the bottom you have base plates. Phage DNA packed inside the head. So the phage, the DNA is packed inside the head. What else? Lambda DNA, the lambda phage DNA is linear DNA. It is a linear, it's a linear form of the DNA. Okay, linear double-stranded DNA. Now the thing about the linear double-stranded DNA is that it has a cohesive single-stranded ends. They are known as cos sites. You can see that this is a single-stranded DNA. Okay, you can clearly see the not single-stranded, double-stranded linear DNA, double-stranded linear DNA, and at the end 5' terminal of both the strands, they have the single-stranded sites known as cos sites. Okay, cohesive ends. This is 12 nucleotide, approximately 12 nucleotide extension in both the from both the strands of the DNA at the 5' site. So this is, you can clearly see the end of the cos site is 5' phosphate group from both the strands. So this cohesive sites or cos site is it's placed in such a way that it has this single-stranded structure, the single-stranded side of the DNA, which can stick to each other like a velcro. They can bind with each other like a velcro, and that is a possibility. And due to this possibility, they can reseal the nick and can form a circular DNA. So formation of circular plasmid is possible here. Formation of circular DNA is possible like plasmid. So when the cohesive sites join together, then we see a structure similar to that of the plasmid like vector. But in this case, this vector has a larger carrying capability, that is, it has a larger insert size capability. So the insert size is 35 to 45 kilo base pair, which is quite big compared to the plasmid. Plasmid can only hold up to 10 to 15 kilo bases, while this phage vector can hold up to 35 to 45 kilo bases. Okay, and now we are going to talk about the different properties, the properties of the phage vector. And property says, you can see that in this particular picture, you can also see after the joining, you can see that there must be this region for head genes, tail genes, means the genes that will be coding for the head part of the phage. This is the gene coding for the tail part of the phage. Then there are these cos sites or cohesive sites. There is this promoter region, and there are some genes responsible for the lytic lysogenic switch. So basically the lambda phage has two different kinds of life cycle, the lytic cycle and the lysogenic cycle. The lytic cycle is the ones where the lambda phage infects a bacteria, and then it imparts the process of cracking the bacterial cell open at the end, known as lytic cycle. Lysis of bacterial cell is done. And on the other hand, what we have is a lysogenic cycle, where once the bacteria is infected by the phage genome will be now recombined with the bacterial genome and stays there for a while, known as lysogenic cycle. And this lysogenic cycle can shift back to the lytic phase whenever the bacteria find the environment feasible to get feasible as to come out as a phage particle, and then the process can repeat. So if we talk about the phage vector properties, the DNA can carry large DNA fragment. This is one important good property. And there are two types of lambda phage, or two types of phage vectors that are available. One is known as insertional vector, and another one is known as the recombinant vector. So replacement vector, the insertion vector and the replacement vector. The insertion vector contains one restriction site, and the fragments up to 18 kilobases long can be cloned utilizing this insertional vector. While the replacement vector, why the term replacement? See, the phage as a vector or as a delivery system of a target gene. In the phage, we have different section of the gene that is responsible for making the head region, tail region, different regions in the gene responsible for making different components of the phage. Now, there are sections of the phage which is not needed by the bacteriophage to spread and transfer the target gene inside the host, which is E. coli in this case. So what we can do is that we can either insert the target gene of interest directly, and in that case only 18 kb maximum capability will be transferred. While if we use a replacement vector, what we do in this case is that it has two restriction site. One is for the removal of stuffer sequence. So basically what happens is that in this whole phage, after joining to the cos site, the circular DNA it forms. Then there are some section which is not needed. Some portion of this DNA is not needed for the process of gene delivery, may be required for other purposes which you don't need right now, because the bacteriophage have multiple functions to play. So you don't need that function, you only need the bacteriophage as a delivery vehicle. So we can extract some portion out. So you can cleave that portion out, known as the stuffer region. It's known as the stuffer region or stuffer sequence. So replace the stuffer sequence with the target or target DNA of interest, and then we can continue the process of gene delivery and cloning. Okay, that's what we can do. Replacement vector is used in this case of transferring the target DNA and replacing the stuffer fragment. Okay, now what are the advantages and disadvantages of the phage vector? You can see the advantages of phage vector. The large DNA fragment can be carried. That is one big advantage, that is 35 to 45 kilobases. We have high gene transfer efficiency. The gene transfer efficiency in this case is maintained is very, very high. That's also important. Third is storage of phage particle is quite easier utilizing the phage vector. Then shelf life. The shelf life can be infinite when you are dealing with bacteriophage, because bacteriophage, as I told you that bacteriophage is a virus. So virus literally is non-living or living. We cannot classify them when it is outside of the host. It's non-living. It's considered non-living. So it can stay for infinite time period. While the disadvantages are out there, that is recombinant DNA may enter lytic cycle and can kill the host cell. That is one thing. The recombinant DNA may enter lytic cycle and can kill the host cell. That is one big problem, because as I told you earlier, that this is a virus, and at intent to kill the bacteria, it always intend to kill the bacteria via the lytic cycle. So it will always try to do that. What we can do is that we can prevent this process. So the recombinant DNA sometimes can get back the feature of killing and causing the lytic cycle to crack open the bacterial cell or host cell. So host cell death is a possibility if somehow the phage receives or retrieves the function of lytic cycle. That's one possibility. And second disadvantage is that the narrow host range. The bacteriophage cannot be used to transfer the gene into any distant related organism. Let's say there's the E. coli as a host organism and there's a bacteriophage. The bacteriophage lambda infects E. coli naturally in the environment. So we can use lambda phage to infect E. coli. So you can take E. coli as a host cell. We can take lambda phage as a delivery vehicle. We cannot use that same lambda phage for, let's say, another bacteria or another host. So host range specificity is another limitation in case of the phage vector. And the third and the last thing is the difficulty to isolate. It's difficult to isolate this because obviously the isolation follows the isolation protocol of a virus. So that isolation protocol of virus and viral culture is completely different thing, and that is totally different from that of the bacterial culture. So that's why we are working with multiple domain. We are working with the virus, we are working with the bacteria as a host, we are working with the gene that is target gene of interest from a eukaryotic cell. So while you are working with a phage vector, we are working with all these different domains. We are working with eukaryotes like human, we are working with bacteria as a prokaryote, and we are also working with virus. So that is one big problem, difficulty to isolate. That is always present there as a disadvantage. But so you can see the phage vectors or use of phage vectors in the process of molecular cloning can be advantageous and disadvantageous. Now depending on the advantage and disadvantage, pros and cons, we can decide whether we'll go with that or not. The biggest advantage in this case will be the large insert size. Other than that, rest of the things are not good. So you can continue that. So with this large size, insert size, if we tag the usability and the easy accessibility or utilization of plasmid vector along with phage, then combining them together, we can achieve both the best part of a plasmid vector and the phage vector together. And that's why we design what is called as phagemid. So where we have best thing from the plasmid, we have best things from the bacteriophage. We combine together, we get phagemid. So phagemid vector is another vector which we will use. We'll see later times, we'll discuss about that. The phagemid, basically we also call it as a cosmid. So cosmid or phagemid, whatever you say, cosmid is what we have. We have the plasmid, the properties of plasmid. Along with that we have cos sites. So plasmid plus cos sites will be cosmids. So now we'll see what cosmids will do. So if you like this video, please hit the like button, share this video with your friends, and subscribe to this channel to get more videos like that in future. Thank you. Bye.